Okay, Bio 121. Now we're coming to the last macromolecule. It wasn't even listed on the first slide. And these are nucleic acids, and they constitute the genetic material of living organisms. And there's long stories about where the names come from and all of this sort of stuff and who discovered them. And the reality is this, is at one point in time we thought proteins were the most important structure, and until we could figure out a way to find a smaller structure or understand that information can be stored, in this case, a double helical structure, two sugar phosphate backbones with a series of nucleotides that share a specific type of bonding. And that physical structure is where we're storing information to build everything we've been talking about. Those carbohydrates, what to do with them, how to process them. The lipids, how to build them, where to build them, when they're being built, how they're being built. Those proteins, the machines that are required to do all of this work I'm trying to mention to you and make it just seem like it's just so simple. But we have two major types of nucleic acids. There's another one we should throw in here, and I'll introduce it at the end. But of course the idea is that if nucleic acids are storing information, you're going to find them inside the nucleus of eukaryotic cells. You're going to find them as a circular strand of information inside the cytosol of prokaryotic cells. Each mitochondria inside of a cell has its own genome, and this is why, at least up until last year, it was thought that all mitochondria were passed from mother to child. It turns out that roughly 11% of the population has mitochondria from their father, not just their mother. Likewise, chloroplasts and mitochondria are these substructures, subcellular organelles, that we believe we picked up, at least inside eukaryotic cells, about 380 million years ago, and they have their own genome. They have their own set of instructions to do what they need to do. And what they do, at least inside of our cells, is they utilize intermediates of glucose breakdown or catabolism, right? And then they use the oxygen to make sure it goes to completion. So they do a lot of things, and ultimately it's taught to you as though you need to know DNA and RNA, even throw in now ATP, because ATP, adenosine triphosphate, is in fact a nucleic acid. So why do you care? Well, you should care for a lot of reasons, but sorry about that. The roles of DNA, of course, they're the codes of the genome for all the entire genetic content. We store it in a specific shape. When it's not undergoing duplication and or transport, it's found in its nascent state as chromatin, okay, which is a complex of DNA and histone proteins. And then chromosomes are thread-like structures that are wrapped around, okay, in a way that, how do I say this? In a way that will allow for easy transport. And we'll come back to this again. DNA codes for thousands of genes. We're talking a minimum if we think just about the human genome, right? Just the human genome. Human genome has, let's just say 24,000 genes, right? And let's just imagine each gene replicator is responsible for storing the information for one protein. So that would mean that you have about 24,000 proteins, but it doesn't work like that. What you're going to discover, and what's going to be really important for you as you take into bioinformatics, is that you can differentially transcribe and translate this so you have multiples of those 24,000 genes to make 24,000 proteins, meaning more proteins, and then each of these will only be specifically functioning in a unique cell type, which means they're in a unique tissue type, and you can begin to see where differentiation and complexity have arisen from the information that was stored inside of your cells, and we just blithely call DNA or genes. Now, one thing I want to make sure you understand is that genes are found within DNA. DNA is the structure that stores the actual coding information, and that coding information will make that mRNA I was talking about earlier. DNA controls all the cellular activities by turning on or turning off genes. And this is well above our pay grade right now, but to understand how this happens, If you're working inside of a room, and imagine that room would be the information you need when you turn on the lights. It makes it easier to get that work done. You shut off the lights. You won't be able to do the work if there's not enough resources to make the work happen. Maybe not a great example, but hopefully you get the idea. Now there's roles for RNA, and primarily our textbook focuses on its role in protein synthesis. And I've worked enough on this stuff. There are many more types of RNA. Every textbook sort of they're like, we can't make it too hard for students to understand. But these structures, these are single-stranded structures where DNA is double-stranded. These single-stranded structures, just their shape alone, is responsible for being involved not only in protein synthesis, but also the regulation of everything that happens after that as well. Messenger RNA, I'm going to teach you in multiple painful ways, is an intermediate nucleic acid that are the instructions for protein synthesis. And then we're going to use transfer RNA that's going to be made inside the nucleolus to they actually bring the amino acids to that mRNA. And then through a series of dehydration synthesis reactions called translation, we now add them together, and that's how we build the primary structure of that protein that then becomes the secondary structure, and then in the correct environment, it maintains its tertiary structure to be functional. Ribosomal RNAs, these tend to be lost on students, but I really want you to understand something. Let's imagine for a second that this is the size of a virus. The size of a virus. Okay, remember, viruses are non-living structures. Yes, no, maybe. And let's imagine this is the size of a cell. Let's imagine this is the size of a cell. Okay, and now let's imagine this is roughly, let's change colors to make sure you get the importance here. This is roughly the size of a ribosome. These things are insanely large structures that are the location where translation will actually occur. And what I'm trying to get you to understand here is that Mother Nature probably underwent some form of ability to build materials at unique locations inside of a cell if the materials were made possible, meaning that mRNA was brought to it and the amino acids were inside the cytosol of the cell and the instructions were found inside the DNA. And to think about how big this is, because it's really important to think about. If you think about how Mother Nature is going to do this, bring amino acids to a specific site, she has to have a location where this can happen and it won't be missed. And by placing them all over the cell, you can begin to see that what Mother Nature is doing here is she's ensuring that you're making the proteins where they need to be made and how they need to be made. Okay, cool. Okay, let's finish this up. Now, there's a difference between DNA, okay, that we sort of draw as double-stranded sugar phosphate backbone structures, and the information you need to know for types of exams. Now, DNA and RNA are nucleotides because of the monomers that are actually making them. What do I mean by that? Nucleotides consist of three parts. You're going to have a nitrogenous base, a pentose sugar, and one or more phosphate groups. Notice the way I've been trying to tell this to you, as I said, it has a sugar phosphate backbone and nitrogenous bases that are interacting with one another. The two families of nitrogenous bases are pyrimidines and purines. The easiest way to remember this is A's bind to T's, G's bind to C's, and there are never any exceptions to that. The reason why this uracil is here is because if DNA is double-stranded, RNA is single-stranded, and that single-stranded structure has uracil in place of thymine. You'll see what we mean as we go along. Now, how does Mother Nature tell them apart? What's the difference between DNA and RNA? Well, I just gave you one clue. RNA is single-stranded and it will have uracil. DNA is double-stranded and will have cytosine, thymine, adenine, guanine. That's not to say that RNA only has uracil. Remember I said uracil in place of thymine, which means RNA has cytosine, uracil, adenine, guanine. Now, The other way that Mother Nature tells them apart is, as I said a moment ago, DNA is double-stranded, but DNA also has a specific type of pentose sugar. It's a deoxyribose sugar, whereas RNA is made up of ribose sugars. The base pairing comes from hydrogen bonds that allow for thymine to bind to adenine, adenine to bind to thymine, guanine to bind to cytosine, cytosine to bind to guanine. And importantly here, notice that the thymine and adenine bonds are 2 hydrogen bonds. Guanine and cytosine are 3 pairs of hydrogen bonds. Now, we tend to lose a lot of students here. And that's because DNA has 2 strands. And imagine, if you will, they're both information storing, which means this cytosine, this adenine, is storing the mirror image of the information that's stored by thymine and guanine. How does it do this? They run anti-parallel to one another. One strand runs 5' to 3', the other one runs 3' to 5'. So here's one strand, 5' to 3', the other is 3' to 5'. This is where somebody always raises their hand and goes, but what if we just turn it around? And if you just turn it around, you're still completely correct, because remember, the information is a mirror image. Now, we're finishing up this part, right? And DNA can express a particular gene by synthesizing unique RNA to undergo transcription, like I said, to complete transcription. That RNA base sequence, of course, it would be slightly different. If DNA is AATTGCGC, the RNA complement to that, you'll see how and why this happens, is CGCG. Okay, so how did that happen? Remember, I told you Mother Nature needs to make some way to distinguish the difference between DNA and RNA. So in the process of transcribing this, let's go A, T, T, G, C, G, C. What's going to happen during transcription? We're going to make the pre-mRNA structure. Notice I changed the vocabulary words there. The pre-mRNA structure is now going to be the complement of that for a single-stranded structure. That single-stranded RNA will have a ribose sugar backbone, so it's ribose, so that sugar phosphate. Sugar is ribose. So that means we're going to place a uracil where a T would have been. A uracil where a T would have been, adenine, adenine. We're going to have cytosine, guanine, cytosine, guanine. And this is how the cell knows that this is now actually RNA. And so once again, it's not just can you remember that DNA is double-stranded and that you're going to make a complementary sequence that's going to replace the U's for the T's. It's going to be, when would this be done? And why is Mother Nature doing this? And imagine, if you will, I give you many more possible sequences you might actually have to maybe work in the other direction. Imagine if I gave you the mRNA complement structure here, and I've asked you to tell me what DNA sequence did it arise from. Can you work this backwards, in other words? Okay, cool. So in this last couple of slides, RNA and the products, right? So what you're going to see is that ribosomal structure I made a big deal out of a moment ago, these actually have catalytic activity, and they're going to allow us now to make specific amino acids in the correct location at the correct time. And here's a sort of model for how it works, where if here's a ribosome, you're going to learn there are 2 parts to a ribosome. There's a large subunit and a small subunit. And what happens is, if here is our now mRNA, notice I changed it from pre to M, pre-mRNA to RNA. And here's our 5' end to our 3' end. I'm trying to give you some orientation. And I want, because they didn't do it here for you, I want to make sure you understand we're moving in this direction. So our mRNA arrives at our small subunit, our large subunit sits down, and what happens is the tRNA, now that transfer RNA, begins to bring in unique amino acids, bam, attaches them, this grows, and now you let go of that tRNA. Will now go off and bind another amino acid, bring back over here. You're going to see they've tried to color code it for you. So you can see that this brought in that unique amino acid through a series of dehydration synthesis reactions. You continue to grow that chain. So DNA, here's a really nice summary in terms of like if I'm going to, in fact, I probably should get you into the habit of the end of every single chapter coming up with some way. In fact, I used to do that. You know, I might already have a solution to this problem. You take your exams, I might have you fill in a series of tables and you have to send them to me. Okay, so this is a great way to summarize information for yourself, whereas this is really general and borderline useful. Function, location, structure, sugar, pyrimidines, purines. For DNA versus RNA, this is a way for your brain to sort of access fragmented pieces of information. Useful for building the general idea. The types of questions you're going to get, though, maybe 40% of your questions would be sort of, do you remember that DNA is a deoxyribose sugar as opposed to a ribose sugar? That should be capitalized. But then, when would you use it? How would you use it? Those are the types of application questions you will get on an exam from me. Okay, so now this gets to the central dogma of life. After looking and looking and looking and looking and looking and looking and looking, you know, people finally put together this idea that once we got beyond the structure of proteins, and we had these things that are called nucleic acids, and the information is actually stored in DNA. Notice that there are two pieces of options that could happen here. The information can be transcribed so we can make a protein, or it can be replicated so we store that information. Okay? And this is a very, very important, not only idea, but very important experimental design question, right? We haven't really thought about experimental design except inside lab. And what I want you to do is think to yourself, well, how could I have proven it worked this way? And that's the general trajectory of this class. Can you think of how the development of biology has changed more over 40 years? I mean, if that's 1970, Crick, Watson, and many others. I'm trying to think of all of the physicists who were actually working on this at the time. I mean, if you think about, well, none of the names you're ever going to know. Maybe you'll know Rosalind, but the idea, of course, is that it's been many years in the making, and it required building specific tools for us to actually begin to see how this was actually happening. So that's the end of chapter 3, and by now, you know, we've gone from what is life, what are feedback systems, what is chemistry inside chapter 2, and this really basic idea that chemical reactions between elements allow us to build materials. And inside chapter 3, what we've done is we've pretty much focused our energies on building the 4 major macromolecules, and those were carbohydrates, lipids, proteins, and nucleic acids. And at the end of the nucleic acids, we finally figured out how they worked. You're going to see that even though DNA is double-stranded, that double-stranded structure has 2 possible fates at any given moment. That information can be transcribed into a single-stranded RNA structure, which will then be translated at those ribosomes to make unique proteins that all have their own characteristics. Likewise, or instead of this process here, to save those instructions, that information can be replicated and stored in this double-stranded structure in the next cell that would use it, or the same cell. Because it's being replicated, it's happening inside that one cell. Okay, that's a pretty good place to stop. Chapter 3 is really thick in terms of details, and you want to figure out a way, like I was suggesting, to break it down into tables for yourself, and then begin to go back and now sort of reassemble it in terms of utility. Okay, bye.